Hello, everybody. My talk here is about the BCG vaccine, which is commonly referred to Bacillus calmette-guerin. Two French scientists who were working on bovine tuberculosis, and by doing continuous passages of virulent strains of M. bovis from 1908 to 1919, about 230, 240 passages, they end up with an attenuated strains of M. bovis that does not induce disease in animal anymore. And later on, some paleobiology studies demonstrated that BCG is a kind of result of large deletion of piece of gene that occurred in the lab during these successive passages, following natural deletions of piece of genes that occurred starting 15,000 years ago. So basically, M. bovis BCG is a close parent of the virulent strains of tuberculosis, which was discovered in mummies in Egypt for like 5,000–10,000 years ago. So these BCG strains obtained in 1919 was first used in human as anti-TB vaccine in 1921, very quickly. So since then, more than 1 billion doses has been injected to humans, and despite this massive vaccination, so currently BCG is still used, and about 100 million doses are administered to newborn worldwide. So despite this massive utilization of BCG, TB continues to be a global threat. About 4,500 deaths every day are counted by the WHO. And most cases of TB are occurring in areas where BCG is used routinely. So BCG continues to be used despite low efficacy because it still protects children for disseminated TB, especially meningeal TB. But it has, as I said, very low efficacy in adult pulmonary tuberculosis. So clearly, and it has been repeated in many reviews, in many audiences, that we need urgently a better TB vaccine. There are too many options, and there are plenty of vaccines currently under development, at least a couple dozens. And one of the options is reshaping the current BCG for the simple reason that BCG is safe. As I said, it has been administered more than 1 billion times without any major side effect, but it's still a good option. So to modify BCG, first of all, at least in my lab, we want to know why BCG is not working. Because so far there is no immunological explanation for the low efficacy of BCG. So classical approach is to look at the host pathogen interaction, see what happened within infected host cell, which is the macrophage. Doing this type of approach, using, working with BCG as an MTB, as M. tuberculosis is the pathogen, using BCG as a surrogate pathogen, many labs, including ours, we found that BCG stops the maturation of the phagosome. After phagocytosis, BCG sinks inside the vacuole called phagosome, and usually this phagosome undergo a process of maturation and then into fusion with lysosomes. and intracellular killing. But it seems that the attenuated vaccine BCG mimics the feature of MTB, the virulent bacteria, in blocking phagosome maturation. And in terms of antigen present in cells, the macrophage needs this step of phagosome maturation prior to present antigen at the surface. And presentation of antigen at the surface for extracellular pathogen, existing pathogen, occur principally via MSC class 2 directed presentation at the surface, and accessory also using class 1 for CD8 stimulation. So here too, we found, and in connection with the first phenomenon, we found that BCG inhibit at least partially the surface expression of MSC class 2. So we've done very simple experiment, like confocal microscopy, to see what's happened at the level of the macrophage, using a cell line, THP1 cells, as a model of macrophage. The reason we use THP1 is that they have very low basal level of MSC class 2, so it can be induced like in physiological condition with interferon gamma. And then we compare what happened with infected cells. So this is the control interferon gamma stimulated cells, and you can see MSC class 2 molecule at the surface. When we infect with killed BCG and stimulate with interferon gamma, we still have surface export of MSC class 2, and we have co-localization of the bacterium with MSC class 2 molecule, okay? Which means that there are sort of MSC class 2 loading with the bacterial antigen. In contrast, when you infect with live vaccine stimulated with interferon gamma, MSC class 2 stay inside the cell. There is kind of sequestration, intracellular sequestration of MSC molecule, and little or no colocalization with the bacterium. And we confirmed this data by showing that infection with BCG has no effect on cell signaling and MHC class 2 gene expression. I mean, cell signaling in terms of STAT1 phosphorylation, which is the major signaling molecule for interferon gamma. And here you have the RT-PCR, kind of semi-quantitative RT-PCR for the alpha chain of DR molecule. And you can see that interferon gamma and infected cells express similar levels. So we conclude that we have intracellular sequestration of MSC class 2 molecule. So during the maturation of MSC class 2 molecule, there are plenty of enzymes involved in the processing. One of them, major enzyme involved in this process, is catepsinase, and we focus on this protease, and we found that in cells that are infected and then treated with interferon gamma, we have reduced catepsin activity. And we found by Western blot reduced level of protein, and by RT-PCR reduced level of gene expression. So the conclusion from here is that BCG infection is reducing catepsinase activity in the macrophage. And by doing so, blocking the maturational processing of MSC class 2, which involve in late stage before surface export, degradation of the invariant chain. Without degradation of invariant chain, majority of MSC molecules stay sequestrated inside the macrophage. So based upon that, we said, okay, let's correct this problem induced by the vaccine. Let's compensate for the inhibition that is produced by BCG. In other words, we plan to deliver with BCG an active form of cathepsinase. So BCG is going to inhibit the endogenous cathepsinase, but by itself deliver an active form. So we choose a vector that is used for mycobacterial transfection, and this is the whole cathepsin protein. It's produced as proenzyme, non-active. It needs an acidic pH for activation. So we cut the active site and we fuse this domain to a signal peptide of an antigen, endogenous antigen in MTB and BCG, which is antigen 85. So the chimeric protein is able to be secreted by the bacterium. And it worked. Here we have a confocal analysis of different conditions, control very little cathepsin S, interferon gamma, BCG transformed with an empty plasmid, you see here, and BCG transformed with cathepsin S, and you see massive secretion going even outside of the phagosome. So the fact that we restored cathepsin secretion via this strategy, we also restored antigen presentation in an, at least in vitro model, where we have a T-cell hybridoma that is responsive for antigen 85, and we have here different conditions, and we look at secretion of IL-2 by this hybridoma. So when BCG is secreting cathepsin S, even without interferon gamma, we have a certain level of antigen presentation, which is amplified in presence of interferon gamma. And here is a control to confirm the concept we're talking about. We have BCG wild type stimulated with interferon gamma and in cells that have been injected with recombinant cathepsin S, and we demonstrate that cathepsin S, active cathepsin S, is responsible for this change of phenotype. So we non-published data from now and on. We did some microarray analysis, quick analysis of what makes the difference between the wild type and cathepsin S. And you can see here the profile are completely different in up and down in terms of gene expression. And we start to do some analysis on pathways. We focused on pathway of interest for us, which are the lysosomal pathway. And this is BCG over control, and you can see in green inhibition, and BCG cathepsin S over the BCG wild type increase. The cell death pathway, we have inhibition with BCG and increase with BCG cathepsin S. And those are the inhibitors of apoptosis. Inhibition of apoptosis is increased by BCG, decreased by the recombinant vaccine. And proapoptotic genes are reduced by BCG wild type and increased by the vaccine, the recombinant vaccine, expressing cathepsin S. So we start to focus on apoptosis since we obtained this data based upon many articles describing apoptosis as a very important phenomenon during macrophage antigen presentation, a new form of antigen presentation that has been described as macrophage antigen cross-priming of dendritic cells. So basically what's happened, infected macrophage, if it undergoes apoptosis, it will shed apoptotic vesicles containing antigen that are delivered to dendritic cells, which are then able to activate T cells. So, as I showed to you in the microarray data, BCG is an inhibitor of apoptosis, and here is demonstrated in cells that are treated with the positive inducer of apoptosis, etoposide. When you infect, with BCG before stimulation, you have significant degrees of apoptosis. So apoptosis, very simplified. You have intrinsic and extrinsic pathway. We don't know how BCG inhibits either one or the other one. We know that BCG inhibits NADPH oxidase and the oxidative burst, which is necessary for apoptosis, but is not a solid link. So anyhow, both pathways goes down to caspase 3 and activation of caspase 3, which by itself activates by degradation a PARP and apoptosis, induction of apoptosis. So our strategy here, as alternative strategy to improve BCG, was to use caspase 3, BCG expressing caspase 3. So we designed a new plasmid for expression in mycobacteria in our lab called PJAK, which is similar to in vitro gene plasmid for the gateway strategy. So we prepared 3 versions of caspase: the wild type caspase, a mutant caspase, position 266, and a reversed caspase where the sequence goes from the small subunit, which is here, the pro and the large subunit. So these 2 versions have been described as kind of constitutively active. So 2 minutes. Okay. So here is the Western blot for the caspase expression. Growth curve of the different strains, the wild type, empty vector, and caspase. Cells infected with the 3 versions of caspase, and we see PARP degradation. The apoptosis induced by reverse caspase 3. And most importantly is the in vivo immune response to BCG wild type versus BCG reverse caspase 3. And here we're targeting antigen 85B, and you can see the improvement of the immunogenicity of BCG. So secretion of interferon gamma and TNF alpha and IL-2 means we have polyfunctional T cells. So the summary is that we apoptotic macrophage shed apoptotic blebs to activate antigen-presenting cells for CD4 and eventually CD8 cells. Thank you very much.
Thank you very much. Are there any questions? Have you tested them for protective efficacy against BCG? Have you compared them in a challenge model to see whether they're better for protection?
Yeah, it's planned. It's very heavy experiments, in BC3 level labs, and we need collaborator for that, but it's in the plan. Yeah.
Okay. Yes.
An extension of that question. So, looking at protective efficacy in the mouse models, but also looking in immunodeficient strains, because if you modify BCG, which is known to be a very safe vaccine, then there's potential for leading to more virulent strains. And of course, there's a concern for something like BCGosis in immunodeficient individuals. So an example is, for instance, the Kaufman Listeria strain, where they introduced attenuating factors. So is that something you're planning to look at?
Yeah, anyhow, it's included. After this immunogenicity demonstration, we need to do safety in SCID mice and protective efficacy in the immunocompetent mice. But the expectation is that the bacterium is further attenuated compared to BCG. So we can even, somehow, we can even propose it for vaccination of HIV positive individual. Since we have data showing that a rapid intracellular killing of BCG caspase relative to BCG wild type. So the expectation is that in the SCID mice, we will see a rapid clearance too.
Okay, thank you, Zakaria, and thanks to the other speakers. It is now lunchtime.